Hello, my friends, and welcome back to class. Now, as you may know, I have a special place in my heart for pop culture and how we can use that to teach different scientific concepts. And in this one, we are watching a clip from the 1982 film The Thing, starring Kurt Russell as the titular dynamic character MacReady. And in this scene, they are utilizing a technique called blood typing, which is a pivotal moment in where the characters utilize this basic principle of anatomy and physiology to identify the alien organism as it has infiltrated the Antarctic base. Here is a brief explanation of the scene from that perspective of anatomy and physiology. The first one is that it's a principle. In this scene, the characters are attempting to determine if any of them have been assimilated by the alien creature. They do this by performing a blood test, specifically a blood typing procedure that you'll learn in class. And this scene from the science fiction movie is actually based on science fact, according to Carl Landster, who developed this scientific concept. Blood typing has some utility behind it, as it utilizes the ABO blood group system, which is based on the presence or absence of antigens on the surface of the blood cell. There are four main types of blood: A, B, AB, and O, and each type is determined by the presence and absence of specific antigens, either called the RH factor. And using the combination of the antigens and the ABO blood system will help inform if any of the crew members on the base got infected. Because it's a dynamic biochemical reaction of the RH antigen present on red blood cells. Individuals can either be RH positive or RH negative. In this case, though, the reaction for compatibility lies within the RH factor, as how that is explained earlier in the movie, that the alien begins to assimilate its victim. And dramatic change in temperature will speed up the reaction. And doing so, taking a look at how this ABO blood group system in RH also informs anatomists and physiologists the compatibility, as blood typing is crucial for medical procedures such as a blood transfusion. An individual's blood type must be compatible with the donor's blood type to prevent any adverse reactions, such as, turning into the thing, as heat is applied to the blood sample. In reality, it's more about the agglutination factor that typically occurs when differing blood types interact with each other in real life. And this blood typing can help answer important scientific questions. In this case, in the movie, they try to use it as a way to determine who the thing actually is. But here in anatomy and physiology, we just want to be able to better understand how to use this technique as a conceptual understanding of what the ABO blood group system really is. Hello everyone, and welcome back to our online digital classroom here in Human Anatomy and Physiology 2. So this week, we are now entering our third thematic section for the semester, and that theme is called Decoding the Symphony of Lesser-Known Interactions of Body Systems. Now, throughout this course, we took a very interesting approach to learning the second part of human anatomy and physiology. In thematic section number 1, we revisit the fundamentals and how we describe, document, and be able to explain the broadest concepts of anatomy and physiology, and reinforce the skill sets you may have already acquired in Anatomy and Physiology Part 1, or be able to be introduced to a new way to critically think about the human body. In part 2 of this class, we learned about the human life cycle, from the smallest unit of life, the cell, all the way up into old age, and what age does to our bodies on a cellular level by studying tissues, but then also on a structural level by looking at the body systems as they age over time. In this thematic section, we are going to basically clean up a lot of the different concepts that are underlying in both courses. And that is why I like to be able to address this as the lesser known interactions. We're going to start that off by looking at the physiology of the blood typing system, as that is a model of investigation. When we think about blood types, it's all about, maybe we can use it in the healthcare field as a transfusion, but conceptually, learning about blood types is actually more geared towards your experimental design. Blood types change according to that ABO blood system or the Rhesus blood system if you're positive or negative. And so being able to utilize those changes in variables based on measurable quantities, we can then be able to help explain aspects of our cardiovascular system in relation to the connective tissue that we commonly call blood. But as part of this course, I just want to be able to show you maybe some diagnostic tests. And one such diagnostic test we'll encounter in our lab this week is how do we test the interaction between different blood type systems and use it as a way to differentiate blood types, but also determine and answer big scientific questions, such as what is our blood type? So I'm going to actually show you this lab demonstration real quick. This lab demonstration can also be found on Brightspace, and you will be conducting it again in your lab. But I've got some little trays right here. These trays are have little wells into them, little depressions, and they're labeled A, B, and then RH. And there's going to be 4 of them. And then I actually have little vials of simulated blood. And so, as part of being able to model this successfully, I'm actually going to be organizing them 1 through 4. And then using the dropper vial, I'm going to place 1 drop of the synthetic blood in each well of the blood typing slide. I'm going to be doing it for each one. Next, I will take some synthetic anti-A and anti-B and put them in the corresponding wells. This is A, then B, and then this clear one will actually be for the presence of or absence of the RH factor. And then mix them around with some little plastic toothpicks. see what kind of reaction I will get. Now, as I'm working on this, I'm already starting to see a reaction to where the blood, as it's reacting to the anti-A or anti-B antigens in the yellow, blue, and clear vials, it's starting to agglutinate and react to what is found on the blood cells. And we can use the presence and absence of, say, the RH and the reaction to be able to catalog and categorize the different blood types to assist us in answering our research question or hypothesis. And doing so, it essentially makes a model in a small square, because we can then use how it agglutinates, what information it tells us, and be able to change the variables so we can learn more about the blood. If you have any questions about this week's material, please reach out. I am here for you and helping you in your learning journey. You can get a hold of me using email, video chat, or swing by my office. And so let's get learning more about that and decode the symphony of the lesser known interactions of body